This is an AQA GCSE Biology Paper, higher tier, paper 1H from May 2020, and I'm going to be doing a talk through showing you the best tips for scoring the highest marks. This question is about photosynthesis. Complete the word equation for photosynthesis. Key word here is word. Don't do a combination of symbols and words. They will not like that. So remember, photosynthesis, it's a process where plants need carbon dioxide, which they absorb at their leaves, water, which they absorb at their roots, and they use it to make glucose and the byproduct oxygen. Describe how energy for the photosynthesis reaction is gained by plants. So we know that that type of energy is light energy. So where do they absorb that light energy? Well, it's by their chloroplasts. So there's a mark for mentioning light and a mark for mentioning chloroplasts or chlorophyll. Students investigated the effect of temperature on the rate of photosynthesis. The students shone light from a lamp onto pondweed and measured the volume of oxygen produced per hour. So remember, what they're changing is the independent variable, so that's temperature here, and what they're measuring is the volume of oxygen, so that's the dependent variable. Calculate the mean value x. Remember, the mean is the average, so we're looking for the average result here. Make sure that they're all quite similar to each other. If you had a very different value, then that would be anomalous and you'd need to discount it, but handily, these are all very similar. So you add them all up and divide by how many there are, which is 3. And once you've done that, you get a value which is 19.1. The students identified one anomalous result in Table 1. Draw a ring around the anomalous result. So which of these results looks super weird and different? They're all similar. 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 Okay, what on earth's going on here? It's this one here. Suggest one possible cause of the anomalous result. So effectively, we've got way too much oxygen being produced. So what could have caused that? Well, maybe the lamp was a lot closer in that experiment, meaning that the plant photosynthesized a lot more. Maybe it was a lot hotter. Anything that would have increased the rate of photosynthesis, really. You could have also said things like the scale was misread or the pondweed had not acclimatized. How did the students deal with the anomalous result? Well, as you should always deal with them, they did not use it in the calculation. And you can prove it to yourself. Look, they used those two results to find an average, which was 2.1. If they'd include the anomalous result, then that number, 2.1, would have been much higher. Give one factor that students should have kept constant in this investigation. That could have been the light intensity in terms of like the wattage of the bulb. It could be carbon dioxide concentration. It could be the length of pondweed or the pondweed species. Table 1 is repeated below. Why did the rate of photosynthesis decrease from 35 degrees to 45 degrees Celsius? Hopefully that's shouting enzymes to you. So when enzymes become too hot, the enzymes are, don't say killed, they're denatured. So if it's decreasing, it's because they've been denatured. Complete figure 1 using the data from table 1. You should include a label on the y-axis, a suitable scale. We're plotting the mean data from these temperatures and a line of best fit. So remember, luckily you've been given that the temperature is in degrees Celsius on the x-axis. So on the y-axis, it means you need to put mean volume of oxygen. Don't forget your units. Now, in terms of what you're plotting on the y-axis, it's this column along here. So the y-axis is the mean rate of photosynthesis. Don't forget your units. That's in centimetres cubed per hour. Take it directly from the table. Pick a sensible axis. We go from 2.1 up to 44. So let's go 0, 10, 20, 30. Because remember, we need to occupy at least half the graph paper. So that's your y-axis. Okay, now we're ready to plot. We know that we're plotting from 25 to 45 degrees. And we're using this highlighted area here, starting at 25 degrees, which was 33.2. For 30 degrees, the value was 44. For 35 degrees, it was 40.8. For 40 degrees, it was 22. And for 45 degrees, it was 2.1. Because they've asked for a line of best fit, you want to do a curved line here. Let's do a curved line of best fit. What is meant by the term diffusion? Well, it's the net movement of particles from an area of high concentration to an area of low concentration. Figure 2 shows part of a leaf. Molecules of carbon dioxide diffuse from the air into the mesophyll cells. Which two changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? So remember, one factor which affects the rate of diffusion is the concentration gradient. So the steeper the gradient, the faster diffusion will take place, which is why this is an answer here. And secondly, if we know that the carbon dioxide is moving here, then clearly if we increase the number of stomata which are open, it's going to give our CO2 more option to move in. Diffusion also happens in human lungs. Figure 3 shows the human breathing system. Explain how the human lungs are adapted for efficient gas exchange of gases by diffusion. Right. So many things to talk about here. We must explain them. So how are they adapted? They have alveoli, which have, this is the explanation, a large surface area to volume ratio. And remember, that's going to increase the amount of gas exchange. The alveoli have thin walls, which is great because it provides a shorter diffusion distance. There are many blood capillaries. Remember, this is a good thing because they surround the alveoli. Why is that a good thing? To allow quick absorption of oxygen. So in terms of the marking points, I'm going to work out where we're up to. Mentioning alveoli is 1 mark. Large surface area is 2 marks. Thin walls is 3 marks. Allowing the gases to diffuse faster is 4 marks. Many blood capillaries is 5 marks. And to allow quick absorption of oxygen is 6 marks. You could have also mentioned that there is a steep concentration gradient of both oxygen and carbon dioxide. So we're writing lots of specialist terms here. Figure 4 shows a root hair cell. Name the process by which water molecules enter the root hair cell. We can see these water molecules here. They're moving from an area of high concentration outside of the root hair cell to a low concentration inside. It's water. It's osmosis as opposed to diffusion. Nitrate ions need a different method of transport. Explain how the nitrate ions in figure 4 are transported into the root hair cell. Use information from figure 4 in your answer. So let's change colour. The nitrate ions are here. Just have a look. There's way fewer nitrate ions outside of the root hair cell compared with inside. So this time they're moving from an area of low concentration to an area of high concentration. This requires energy. So hopefully the name of the process being screamed at you is active transport. Figure 5 shows 3 types of cell. Give 2 similarities between the prokaryotic cell and eukaryotic cells. Okay, what do these terms mean? Eukaryotic means they have a nucleus. Prokaryotic means no nucleus. So let's make sure we label these correctly. The bacterial cell is prokaryotic. The liver cell and the mesophyll cell. Look, they both have a nuclei. Well, what can we see that's similar between all these cells? Well, look at these little black dots. What are they supposed to be? Ribosomes. What else can we see is common? Well, the grey shading is cytoplasm, as well as this membrane here. It's the cell membrane. So both have cytoplasm or cell membrane. Give 3 differences between the prokaryotic cell and the eukaryotic cell shown above. So what's different? Well, the most obvious thing here is that the prokaryotic cell doesn't have a distinct nucleus. What else is true? Look at these little circles of DNA. These are plasmids. Neither of the eukaryotic cells have plasmids. You can write a full comparison, obviously. And then what else is different? Well, look at these sizes. They're massive in comparison to this single micrometer bacterial cell. So use the information on the diagram. Don't dig around too much, guys. So prokaryotic cell is smaller. Give the ratio of the size of the bacterial cell to the size of the mesophyll cell, so make sure you're looking at the right one. There's the bacterial cell, there's the mesophyll cell. So it's 1 micrometer to 0.05 millimeters. We've got issues here because the units are different, so get them both into micrometers by timesing millimeters by 1,000. So that becomes 50 micrometers, so the ratio here is 1 to 50. Name the type of cell division that produces genetically identical body cells for growth and repair. There's the clue, growth and repair. Mitosis. Remember, the daughter cells here are genetically identical. Figure 6 shows a cell cycle. What percentage of the time for one cell is represented by stages 2 and 3? So we're just looking at this area here. We'll count how many slices do we have. 1, 2, 3. So there's 20 slices altogether. How many of those slices take place in stages 2 and 3? 1, 2, 3, 4, 5, 6, 7. What's 7 divided by 20 times by 100 to get it into a percent? It's 35%. Describe what happens in each stage of the cell cycle. So stage 1, that's the longest part of the cycle. That is where your organelles replicate. So let's name some organelles. It's also the stage where DNA replication takes place. Stage 2 is really mitosis. So what takes place there is one set of chromosomes is pulled to each of the opposite sides of the cell. That's quite an in-depth answer there to do with how mitosis is actually carried out. And then lastly, stage 3. This is the proper division stage, so the cytoplasm and cell membrane divides to form 2 daughter cells. (4.1) Lipases break down lipids. Which 2 products are formed when lipids are broken down? Well, lipids are broken down into fatty acids and glycerol. Remember, amino acids are the breakdown products of proteins. Glucose is the breakdown product of starch. And glycogen is a carbohydrate, so nothing to do with fats. So try and prove to yourself why an answer is wrong as well as why an answer is right. One model used to explain enzyme activity is the lock and key theory. Figure 7 shows the model of this theory. So we've got the enzyme fitting together with the substrate in order to form these products. Explain the lock and key theory and use information from figure 7 in your answer. So what can we see happening? Well, the enzyme binds to the substrate because they have a complementary shape. And at the end, the substrate is broken down into 2 products. Notice that our enzyme is unchanged from beginning to end. So the enzyme's active site is complementary to the substrate. The substrate breaks down into 2 products. And finally, the enzyme is unchanged. There are many different types of lipases in the human body. Why does each different type of lipase only act on one specific type of lipid molecule? I've kind of already talked about that actually. It's because each of the enzyme or lipase's active site is only specific or only fits only one shape of substrate or lipid molecule. Students investigated the presence of starch and glucose in the leaves of geranium plants. This is the method used. Place 2 identical geranium plants on a bench near a sunny window for 2 days. After 2 days, leave one plant near the window for 2 more days and place one plant in a cupboard with no light. Remove one leaf from each plant, crush the leaf to extract the liquid and test the liquid for glucose and starch. So let's try and work this out. In situation 1, the plant's allowed to photosynthesize as normal, so it's going to be producing starch endlessly. In the second situation, we know that it's in the dark, so it can't be photosynthesizing, so therefore it uses up its starch. Describe how the students would find out if the liquid from the leaf contained glucose. Right, this time you want to use Benedict's reagent and heat in a water bath. If it goes brick red, we know glucose is present. Describe how the students would find out if the liquid from the leaf contains starch. This time use iodine. If it goes blue-black, you have starch. Being nice and specific. You don't need to use loads of words here. Table 2 shows the students' results. So, leaf from the plant kept in the light for 4 days. You're positive for both glucose and starch. Leaf from the plant kept in the light for 2 days and then no light, we're negative for starch. Explain why the leaf in the light for 4 days contained both starch and glucose. The leaf photosynthesized, producing glucose, because remember that is a product of photosynthesis. And then remember, some of that glucose is converted to starch for storage. Because remember, that's an insoluble carbohydrate. Explain why the leaf left in the cupboard with no light contained glucose but did not contain starch. I already mentioned that above. Because it can't photosynthesize, starch is used up and converted to glucose. The plant is unable to photosynthesize due to the lack of light, so no glucose is made, hence the need for converting starch to glucose. Why is the glucose needed? So that the plant can carry out respiration. Because remember, respiration is that number 1 living organism process. Do not confuse respiration and photosynthesis. Suggest one way the students could develop the investigation to find out more about glucose and starch production in plants. Anything sensible here. My favourite one here is test other species of plants. I think that's the most straightforward one. You could also say test variegated leaves, so ones that have dark and light patches. You could vary the light levels, or you could test the roots and stems. Many plants have evolved defense mechanisms. Figure 8 shows part of a gorse plant and part of a deadly nightshade plant. The gorse plant has evolved to have sharp thorns. What type of defense response are represented by the thorns? That's a mechanical type of defense. You could have also written physical or structural here. How do thorns defend the gorse plant? Well, they're sharp, aren't they? So that's going to stop animals or herbivores from eating the gorse plant, and therefore the gorse plant remains whole and undamaged. So thorns stop herbivores, so that's animals which eat plants, from damaging the gorse plant. The deadly nightshade plant has poisonous berries. What type of defense response are poisonous berries? Because they contain poison, it's a chemical response. A scientist noticed that in one area the gorse plants had yellow leaves and stunted growth. One reason for yellow leaves and stunted growth is the deficiency of nitrate ions. Remember that they're needed to build plant protein. Explain 2 other possible reasons for the yellow leaves and stunted growth. Do not refer to nitrate ions in your answer. Okay, so what else causes yellow leaves? Well, that's a magnesium deficiency. Why would that cause yellow leaves? Because magnesium is responsible for making chlorophyll, which remember is a green pigment found in chloroplasts. So less chlorophyll means less photosynthesis. And less photosynthesis means stunted growth due to lack of glucose production. What about the yellow leaf stunted growth from a pathogen point of view? What sort of disease could cause that? Tobacco mosaic virus. Tobacco mosaic virus damages chloroplasts, leading to everything we mentioned above. Less photosynthesis, less glucose, less growth. The gorse plant has nodules on its roots. The nodules are part of the living root tissue. Bacteria which convert nitrogen gas into soluble nitrate ions live in the root nodule tissue. Okay, so that's a great thing. Suggests how the nodules benefit the bacteria. Why would the bacteria be on the nodules? Well, let's say that the nodules provide something that the bacteria needs. Nodules provide bacteria with glucose, which is needed in the bacteria's respiration. Explain how the nodules benefit the gorse plant. Well, this is a good thing. Nodules allow bacteria to produce nitrates. Why are the nitrates good? Nitrates are needed by the plant to build proteins from amino acids. If you hear the word nitrates, I want you to think about proteins. For many years, drugs have been extracted from plants. Which plant material was chewed as a painkiller? It's actually willow. Remember, that's where the origin of aspirin actually came from, salicylic acid. Question 6. Data from the Million Women Survey in the UK was collected over 15 years. Scientists analysed the data to study the effect of consuming alcohol on liver disease. They included 400,000 women who regularly consumed alcohol. They included 400,000 women who did not consume alcohol, and they excluded women who already had liver disease. Age and gender were controlled. Many other factors were also controlled. Suggest 2 other factors which the scientists would have controlled. I definitely think the diet of the woman is important because that could definitely contribute to liver disease, as well as the fitness level of the woman. You don't want to be comparing super fit women with unfit women. You could have also said the medication they're on, their smoking habits, whether they're obese or not, as well as their ethnicity. The data was analysed for women who drank alcohol with meals, for women who drank alcohol not with meals, and for women who did not drink alcohol. During the survey, approximately 1,500 women developed a liver disease called cirrhosis. Scientists calculated the relative risk of developing cirrhosis of the liver for each group who consumed alcohol. A relative risk of 1 means that there's no statistical difference between the groups who did consume alcohol and the group who did not consume alcohol. So that means that 1 means not statistically relevant or important. So down here is important. A woman drinks 150 grams of alcohol per week, not with meals. The woman decides to change her drinking to 150 grams of alcohol with meals. Calculate percentage decrease in relative risk of developing cirrhosis of the liver for this woman. So 150 grams. That's important. Let's read off here and here. So this is no meal, so that's going to be worse for you. This is with a meal. What are these two numbers on the x-axis? On the y-axis, 2.5 versus 1.6. So 2.5 minus 1.6. You want to divide it by the original value, which is 2.5, and then multiply it by 100. So remember, to find out the percentage decrease, you do change over original value times by 100. That's what I'm doing here. So 0.9 divided by 2.5 times by 100 equals, and that equals to three significant figures, 35.3%. Don't worry if you've got a slightly different answer. There's quite a range of answers they'll accept here. 1 glass of wine contains 12 grams of alcohol. A different woman drinks 2 glasses of wine each day with her meals. Calculate the relative risk of developing cirrhosis of the liver for this woman. So that means she's taking in 24 grams of alcohol. And then look, this is per week, so we need to do that 24 and multiply it by how many days there are in the week. So that becomes 168. So let's look up here. Just going to make a note, that's 160 grams of alcohol per week, and it's with meals. So I need to look at the dotted line. It's about here, which makes it about 1.83. I'm going to say 1.8. Figure 10 is repeated below. Consuming alcohol with meals instead of not with meals decreases the relative risk of developing cirrhosis of the liver. Give 2 other conclusions about the relative risk of developing cirrhosis of the liver related to alcohol consumption. Use data from figure 10 in your answer. So you can see here that as our alcohol levels increase, it doesn't matter if you've been having it with a meal or not, you're going to increase your risk of developing cirrhosis. If you just compare this bit of the graph here, as long as you have less than 50 grams per week, it doesn't really matter how much it is. It has no effect on your relative risk with a meal. However, without a meal, you're still going to see a slight increase in your relative risk as your alcohol consumption increases. So the easier marking point here is consuming alcohol increases the relative risk both with and without meals. Let's use some data to support this. Let's just use the one without meals. I'm just using points which are easy to read off. So when you consume 150 grams of alcohol per week compared with 50 grams of alcohol per week for the person that didn't consume it with a meal, you can see that the relative risk increases from 1.3 at 50 grams to 2.5 at 150 grams. So not with a meal, relative risk increases from 1.3 to 2.6. With increased alcohol consumption. I'm really running out of space here. Don't know how on earth they think I'm going to quote data in such a small space. And our second point is consuming less than 50 grams of alcohol per week does not affect the relative risk when drunk with a meal. And what am I on about? It's this bit of the graph here. Suggest 2 reasons why the data is considered valid. Well, validity is all to do with repeats and how many people you've sampled. We were told we had a really large sample size higher up. 1,500 women, yeah. So firstly, the validity is great because we've got a large sample size of 1,500 women. What else can we say about it? Well, again, much higher up. They ran this experiment over, let's have a look, 15 years. Which is amazing, really, as far as the scientific study goes. So just one aspect of the survey which might reduce validity. I think lots of people tend to lie about how much they drink to make themselves feel less guilty. I'm not trying to make judgments, I just think that is something good to mention here. They tend to underestimate. Cirrhosis of the liver tends to lead to liver failure. Describe the effects of liver failure on the body. Okay, well, what is the purpose of the liver? So damaged liver means less bile is made. What is the role of bile? Well, you're going to find that less emulsification of fats takes place. So less emulsification of fats takes place, meaning that their surface area is an increase. What's the second role of bile? Well, it neutralizes hydrochloric acid from the stomach. So less bile means less neutralization of stomach acid or hydrochloric acid in the small intestine. So fundamentally, you're going to find that there's less absorption of food molecules. What else does the liver do? Well, it breaks down excess proteins. So less urea, so less ammonia and urea is produced. And any ammonia that is produced will remain and hang about, meaning that it's super toxic. You could also talk about the liver's function in breaking down alcohols and other drugs and toxins. But yeah, it's worth 4 marks, so be super specific there. Monoclonal antibodies are usually made using mouse lymphocytes. Candida albicans infection, which is actually known as thrush, produces serious symptoms in patients with a poor immune system. Recently, scientists have produced monoclonal antibodies to Candida albicans using human lymphocytes produced naturally after infection. Candida albicans lives in the throat of infected patients. A sample is taken from a throat of a patient with suspected infection. The sample is transferred onto a microscope slide. Describe how the monoclonal antibodies and the fluorescent dye could be used to see any Candida albicans pathogens on the slide. So how is this going to work? Well, first of all, we use that dye in order to identify the antibodies. What do the antibodies attach to? Well, they'll attach to the antigens on that Candida albicans. So the monoclonal antibodies attach to antigens, which remember is the marker on the pathogen. The monoclonal antibodies have the fluorescent dye previously attached, meaning that the C albicans can be identified under a UV lamp. You're using a UV lamp because we've been told that the dye is fluorescent. In a laboratory, the human lymphocytes, monoclonal antibodies, were injected into animals infected with this Candida albicans. The monoclonal antibodies increase phagocytosis. That's good. Remember, that means that the pathogens will have been engulfed by white blood cells. Doctors intend to start a trial to give the monoclonal antibodies to patients severely ill with Candida albicans. Explain how increased phagocytosis of the Candida albicans pathogen will help the patient. Don't stress about all these words. It's saying, why is phagocytosis of the pathogen a good thing? Because it means that more of the pathogen will be killed by phagocytes. Why is that a good thing? And so there's less damage to cells by the pathogen because it's not there anymore. It has been shown that this monoclonal antibody treatment is effective in the laboratory using both infected tissue culture cells and infected live animals. The monoclonal antibodies treatment for Candida albicans is now ready for clinical trials on people. Describe how the clinical trials should be carried out. So, drug trials. Well, remember with the clinical trials, first of all, we need to try it out on healthy volunteers, followed by people who are infected, with the Candida albicans. And remember, we're looking for things like efficacy, dosage, toxicity. So these are all things we need to consider, and it's worth 6 marks. So first of all, test the treatment on healthy, I've already mentioned this, healthy volunteers, and you need to use a very low dosage in case it's at all toxic. So to check its toxicity, and we're already at 3 marks here. Then test on patients with the disease. Why do we do that? To test for any side effects and to check its efficacy. So that's how effective it is. Efficacy is a great way to do that. Use a double-blind trial, which remember is when neither the patient or doctor knows who's received the treatment or a placebo. And run the trial for a long duration, so maybe 10 years or so, and with many volunteers. So where are our marking points? I'm highlighting them in orange. Healthy volunteers is a mark, very low doses is a mark, to check its toxicity is a mark. Now testing with patients with the disease for a mark, mention side effects for a mark, efficacy for a mark, double-blind trial for a mark. And this is a mark, so many, many points. Not repeating myself, being nice and accurate. Scientists have also used human lymphocytes to make monoclonal antibodies to other pathogens and some types of cancer cell. Suggest one reason why these new monoclonal antibodies have been more successful in treating diseases in humans than monoclonal antibodies made using mice. Okay, well, with mice, you're going to have rejection issues. So, monoclonal antibodies made using humans will not suffer the same rejection issues as one made from mice. Right guys, I really hope you found that helpful. Don't forget to give it a like and comment if you'd like to see similar videos in the future.